Okay, this is the general review for Unit 1. This is not going to be as detailed as the material that's covered in both the pre-lab lectures or in the notes handouts that are available on Blackboard. So before the quiz and exam, it would be very beneficial to review those more detailed documents to be sure that you maximize your ability to score well on the exam. So in Unit 1, we first started looking at the ways in which we classify life. So we start off talking about a taxonomy. So a taxonomy is simply an ordered way to classify organisms, and a taxonomy is arranged in groups that start with very general groups, like the archaea, eukarya, and bacterial domains. And as I've stated before, in this laboratory class, we're going to look at the characteristics of eukaryotic organisms exclusively. So you can see here at each level of taxonomic organization, we're getting more and more refined. So each of these groups are sharing fewer and fewer features until you get down to the species level, where you have the binomial naming convention that was developed by Linnaeus. So the Panthera pardus. Panthera is the genus, and pardus would be the species. So we want to also be able to diagram our hypothesis about the evolutionary relationships between organisms, and we display this on a phylogenetic tree. So each branch point or node represents a common ancestor. So that common ancestor then diverged into different species, and the length of each of the branches represents either the time or the number of changes in a DNA sequence if the tree was built from DNA sequences. And ideally, you want a branch to branch just off into two branches, but there are times where you can have the possibility that you cannot discriminate between the features of multiple organisms, and you diagram this in what we call a polytomy, because it's more than two branches. And branches that are adjacent to one another on the phylogenetic tree are referred to as sister taxa. So in order to build a tree, you need to look at different characteristics in different organisms. So here's an example of a data matrix, and the way you read these data matrices is if there is a zero, that means the organism has the ancestral character, and if there's a 1, that means the organism has the new character, which we call a derived trait. So from this data matrix, you can construct a tree. So we have an outgroup here, and the outgroup will have all ancestral characteristics. So then we can use this information to build a tree. I'm going to try and cram it in the corner here. So we have the lancelet, which would be the outgroup, and all of the other organisms have at least one of the derived characters. So then the first derived character is the presence of a vertebral column. So all of the organisms have a vertebral column. But the lamprey has a vertebral column but no hinged jaws. So all the other organisms have hinged jaws. The tuna does not have 4 walking legs. Whereas all of the other organisms do. And then the salamander has 4 walking legs, but does not have the amnion or does not have the amnion or hair. So, whereas the others have the amnion and hair. This would be salamander. And then finally, we have the turtle, which has an amnion but no hair. And the mammals, or the leopard, which is a mammal, which has hair. So you can mark all of the characters that support these branches. So here we have a vertebral column. Here we have the jaw. Here we have 4 legs. Here we have the amnion. And here we have hair. So from this information, we can build a phylogenetic tree. So one important feature in the evolution of protists is the formation of plastids, which allow for photosynthesis. So it's believed that an ancient eukaryote engulfed a cyanobacterium. So cyanobacteria are photosynthetic bacteria, and this formed an endosymbiosis to form the chloroplast. And this first time the cyanobacterium was engulfed, so when a prokaryote was engulfed by a eukaryote, we call this the primary endosymbiosis. Okay, but at multiple times in evolutionary history of the dinoflagellates, eukaryotes engulfed either red or green algae. So a eukaryote engulfed another eukaryote. So that's why we call this secondary endosymbiosis. So this has happened where a red alga was engulfed, and this led to the type of plastid that we see in the dinoflagellates, the apicomplexans, and the stromenopiles. And then we have other examples where a green alga was engulfed, forming, again, another secondary endosymbiosis because you have a eukaryote engulfing a photosynthetic eukaryote. And this led to the origin of the euglenid family and these other organisms that we really didn't talk about, the chloractinophytes. But we don't really need to concern ourselves with those because we didn't cover them. So you need to be able to recall the supergroup that all of these organisms are in. So we have 5 supergroups. So we have the excavata, the chromalveolata, Rhizaria, Archaeplastida, and Uniconta. So we're going to go through each of these supergroups one by one. So the supergroup excavata includes the organisms Euglena and Trypanosoma, which we looked at. So Trypanosoma is a diplomonad. Whoops. So Trypanosoma is a diplomonad, and Euglena is a euglenozoan. And as shown on the study guide, for each of these organisms, you should know their supergroup as well as the structure of their cells, the mode of nutrition, are they autotrophs, heterotrophs, or mixotrophs, and for those that we covered, the life cycle. So this is a diagram of Trypanosoma. So it's a blood smear, and you can see the kind of corkscrew-shaped body and the thin flagella extending off of it. The Trypanosoma are unique in that they have a special large chloroplast with a massive DNA in it that's called a kinetoplast. And this organism is transmitted by the tsetse fly, and as we went over in lecture, it requires that tsetse fly host to complete its life cycle. So you may want to review that in the notes. So the euglenozoans, or Euglena, is a photosynthetic protist that has a nucleus, and it moves using a long flagellum. It does have an eye spot. In the slides that we have, it's very difficult to see. Really, only the nucleus was very clear. And they have a special cell wall called a pellicule, which is, as you can see, kind of these spiral strips of cellulose that are protecting the cell. The next supergroup was the chromalveolata, which include two main subgroups. We have the alveolates, which include dinoflagellates, the apicomplexans, and the ciliates. And then we have also the stromenopiles, which include diatoms, golden algae, and brown algae. We only looked at examples of diatoms and brown algae. So this is the life cycle of Plasmodium. It's the causative agent of malaria. So it is transmitted via a mosquito host. So the mosquito bites an individual, and the parasite enters the bloodstream in the saliva. Those sporozoites go to the liver, and inside the liver cell, they reproduce to make these cells called the merozoites, which then go on to infect red blood cells. And the cells that are released are gametocytes. And if an individual is bitten by another mosquito, then those gametocytes enter the gut of the mosquito and develop into gametes. You get a zygote, which forms this oocyst, which forms new sporozoites, which migrate into the salivary gland, and the transmission cycle will continue. Another group in the Chromalveolata are the ciliates, Paramecium and Stentor. So Paramecium can reproduce either by binary fission, in which the cell just divides, grows and divides, or it can grow, it can reproduce sexually by the process of conjugation, which is diagrammed in the cycle below. So the ciliates have two nuclei. They have a large macronucleus and a smaller micronucleus. The micronucleus contains a single copy of the genome and will undergo meiosis to form four haploid cells, four haploid nuclei. Those nuclei, three of them degenerate, and the one remaining nucleus undergoes a mitotic division. So now you have two genetically identical micronuclei, and the conjugating cells swap micronuclei in a process similar to fertilization. Those nuclei fuse into one nucleus, and then they begin to divide, and the nuclei fuse to form a new macronucleus, and the old macronucleus degenerates. So now you end up with two genetically unique or distinct cells. Another group in the Chromalveolata are the dinoflagellates, which is diagrammed here. So the dinoflagellates have these cellulose plates or shells, which kind of overlock and interlace. And dinoflagellates, they get their name move by the use of flagella, which you can see coming out between the two halves of the shell. And dinoflagellates are photosynthetic organisms and are responsible for red tides and are a major part of the phytoplankton in marine environments. So moving on to the stramentopiles. So diatoms are stramentopiles. So they're photosynthetic organisms, and their cell walls, in contrast to the dinoflagellates, are made of silica or glass-like material, and they are among the most important food sources in marine systems. And as you can see, they've got a large diversity in the shapes of their cell coverings. The other group of stramentopiles that we looked at are the brown algae, or commonly referred to as seaweeds. So they have particular structures. So many of them are anchored to the rocks. So you have at the base, you have a holdfast, which is anchoring the alga to the rock. Then you have the stipe, which is a stem-like structure, and then you have leaf-like blades. The next supergroup are the rhizarians. So the two rhizarian groups that we looked at are the foramniferans and the radiolarans. So the foramniferans have a calcium carbonate shell, or test, T-E-S-T, is another common name for the shell. And you can see that the test is perforated, so the organism can extend pseudopods through those pores, which can be used for locomotion and feeding, so food particles will attach to them. And another important or interesting thing about these foramniferans is that they'll form a symbiosis with cyanobacteria in their shell to acquire extra nutrition. But it is not an endosymbiosis. They're free living outside of the cell. The radiolarans extend through their porous shells actinopodia, which are very slender pseudopods, and those pseudopods, like for the foramniferans, are used for feeding and locomotion. And the shells, in contrast to the foramniferans, the shells of these organisms are made of silica, much like the shells of the diatoms are made of silica. Okay, the next major supergroup are the Archaeplastida. So not all Archaeplastida are classified as protists. Only the red algae and the green algae, and there are two types of green algae, the chlorophytes and the charophytes, are classified as protists. The land plants are not classified as protists. So examples of red algae are porphyra and corallin algae, and the green algae would be Volvox, Chlorella, Spirogyra, or not listed here that we looked at, Chlamydomonas. Let me go back here for just a second. So some of the key features here of the red algae, they get their name because of a red pigment, phycoerythrin, which helps them more efficiently do photosynthesis in the deep tropical waters where they are found. And green algae are named obviously because of their green color, and they get that from the chlorophyll pigments that are found in the plastid, in the chloroplast. And all of these organisms have plastids that are derived from the primary endosymbiosis, so that's why they are referred to as Archaeplastida, or ancient plastid. Okay, the next organisms that we looked at were the uniconts, and the protist uniconts are the slime molds, gymnamoebas, and entamoebas. And later we will look at both animals and fungi, but those are not classified as protists. Okay, so this is a table showing some of the different supergroups and subgroups within those, and their key characteristics. So this is a useful tool for learning, the useful table for learning the characteristics. But certain things on here, such as the nuclearids and choanoflagellates, we did not look at. So you only need to concern yourself with the groups and the organisms that we looked at. So the other major group that we looked at in the Archaeplastida were the non-vascular land plants. So we looked at two different plants. We looked at Neem, which is a moss, and Marchantia, which is a liverwort. So this is an example of a moss. This is sphagnum moss, but you can see its structure is quite similar to the Neem. So at the base, they're anchored to their substrate by rhizoids. And the dominant generation that you see in the environment is the gametophyte. So you have a stalk with leafy structures coming off of it. And then at the tip of that leafy stalk, you will form either antheridia that produce sperm cells by mitosis, or archegonia, which produce egg cells, again by mitosis, because the gametophyte is haploid. Okay, and then after fertilization of the egg cell, you will form a sporophyte, which is this structure here, which has at the base a foot, then a stalk-like structure called the seta, and then finally the capsule. Okay, and in the capsule, cells are going to undergo meiosis to form spores. Okay, the liverwort, again, has male and female plants. So the female gametophyte has this umbrella-shaped structure where you form the archegonium with the egg cells. And in the male gametophyte, you form antheridia with sperm cells. And you can see in the female, the archegonia are on the underside of the cap, whereas in the male, the antheridia are on the upper surface. Okay, and an important feature of plant reproduction is the alternation of generations. So plants alternate between a haploid gametophyte, which is going to form gametes, eggs or sperm, by mitosis. Then those eggs and sperm are going to fuse in a process of fertilization to form a zygote, which will develop into the sporophyte, and that sporophyte makes haploid spores by meiosis. That's a key point of the alternation of generations. So the embryo is retained within the tissue of the gametophyte, so that's why we call plants embryophytes, because that embryo is dependent on the parent, although it is an independent organism. So if we look here at the life cycle of Neem, the moss, so spores will be released into the environment. They will germinate to form these filamentous structures called protonemata. Those protonemata will form either a male gametophyte or a female gametophyte. And at the tip of the male gametophyte, you form antheridia, which form sperm cells by mitosis. And in the female gametophyte, you form archegonia, which form egg cells by mitosis. And as I've said in class, the egg cells are going to be the largest cell that you see within that structure. So the sperm cells are going to swim through a layer of water and are attracted to the archegonia and will swim down and fertilize the egg cell to form the zygote. That zygote will develop into the sporophyte, and when fully developed, the sporophyte is attached to the female gametophyte by the foot. The stalk-like structure is the seta, and then at the terminus of that seta, you have the capsule, and inside the capsule is you're going to undergo meiosis to form new spores. And then the life cycle will continue. So the key thing about this life cycle is you've got separate haploid generations, the gametophytes, and a separate diploid generation, which are the sporophytes. So that's the end of the review. So the exam or the quiz will be available during normal class time between 10 and 11:50. And good luck. And as I said, it is an extra credit quiz.